Last class, we started this unit and we started focusing on the difference and similarities between prokaryotic and eukaryotic cells. Remember, eukaryotic cells have a true nucleus and they have membrane-bound organelles. And we went through the advantages of organelles. Organelles allow cells to isolate molecules that should not be interacting. It allows it to enclose and concentrate molecules so the reactions will happen faster. And many organelles are folded, and that increases the surface area where reactions can take place. On this page, we started going through the different parts of the cell. You need to be able to identify them and tell me what they do. What are the functions of these cell parts in cells? And we looked at the plasma membrane and the cytoplasm. And today we're going to continue on. We're actually going to stay right here. We're going to continue on looking at these different cell parts. With your learning targets today, the ones that are in yellow are the ones that we covered first. And now we're going to get into the blue ones. So we're going to be talking about other cell parts. You can see that we're going to look at cell parts that produce proteins. We're going to look at things like the ribosome and Refiar and Golgi and so on. We're going to look at the difference between attached and unattached ribosomes, and we're going to talk about after these proteins are made, where they can end up in the cell. Now your notes from here on out are organized into cell parts that all are working together to perform a task for the cell. So the next group of cell parts that we're going to look at will be the nucleus. We're going to take a look at ribosomes. And ribosomes come in two forms. They're called free ribosomes, or unattached. And then we have the attached ones. We've already looked a little bit at ribosomes because they can be found on the Refiar. So we're going to look at the Refiar. Golgi. The Golgi you probably have heard of before. It can go by the name of just Golgi, Golgi body, Golgi apparatus. We're going to take a look at lysosomes today. And we're going to look at that plasma membrane. Remember, the plasma membrane and the cell membrane are the exact same thing. Plasma membrane. Now, the reason why I've grouped these together is because we're going to see how these interact to produce and transport proteins around the cell. And that's what this image is kind of showing you here, that we're going to talk about the nucleus and how it relates to ribosomes and the Refiar and the Golgi and the cell membrane, and we'll even talk about some lysosomes. So starting with the nucleus. Remember that the nucleus is going to be a large center structure in eukaryotic cells. And we've already talked a little bit about the functions of the nucleus. It protects the DNA. One function is protecting the DNA of the cell. And the DNA is so important because your DNA is the instructions for building your proteins. If your DNA codes for the wrong protein, then you'll have some kind of genetic condition or some kind of genetic disease. For example, the girl that doesn't feel pain. She can step on a nail, it can go through her foot, but she's not going to feel any pain. The only difference between you and her is a protein. She doesn't make a protein correctly, so her pain neurons didn't develop, whereas you made the protein correctly and your pain neurons definitely developed. So your DNA are the instructions for building your proteins. That's why sometimes the DNA is referred to as a blueprint molecule. Blueprints that tell you how to build proteins, whereas blueprints will tell you how to build a house or a barn. Now, the DNA is super important, and we will never let it out of the nucleus. So what we're going to do to the DNA is we're going to make a copy of the instructions. And that's what this diagram is showing you right here. That this right here is the copy, it's a molecule, and we already talked a little bit about RNA. So this is an RNA molecule. As a matter of fact, that M stands for messenger. So we're going to copy the DNA and carry that information, carry that message to the construction site where the proteins are going to be built. And that's what you see happening right here. So here's the mRNA. You can see that it's leaving the nucleus, and it has found the cell part that is going to read it and make a protein. So here's the ribosome, and we can see right here that's a polypeptide chain. So a protein is being built by this ribosome. So we're going to come back over here and say copies of the instructions for the proteins are made, that's that mRNA molecule, and they're leaving the nucleus. They're going to be sent to ribosomes. Now let's take a closer look at ribosomes. Ribosomes can be diagrammed several different ways. You can see that here we really zoomed in on a ribosome, and it's very, very big, and it's actually showing what the ribosome does. Lots of times, though, whenever you look at the image of a cell, we're farther away, and the ribosomes are going to be these little dots that you see. And some of them are free. They're just floating around the cytoplasm, and then some of them are attached on the rough ER. Now, those free ribosomes, we can also call them unattached. And then again, some are attached onto that rough ER. You might circle the first three letters of ribosome, ribs. If we're eating some ribs, then we're going to be getting protein because protein comes from meat. So you might remember ribs, and that'll help you remember protein. And specifically, the job of the ribosome is to make proteins. There's no other way for your cell to make proteins. All proteins are started and made by the ribosome. Now, you're going to be building models here in a little bit, and you're going to have to each explain to me how different cell parts interact. So let's just kind of review the first two that we've seen so far. We've talked about the nucleus, and we've talked about the ribosomes. So how they're going to interact is that ribosome receives a copy of the instructions. Remember that copy is called mRNA, and it's just a copy of one little gene on the DNA. Genes code for proteins. And that comes from the nucleus. So the nucleus holds the codes for the proteins. That code is copied and sent to the ribosome, and the ribosome is going to take the monomers, which are amino acids, string them together, and then you have a polypeptide or a protein. Now, I'm going to play a narration, and it's going to show you what I mean by a copy comes from the nucleus and goes to an attached ribosome. And then it's going to take us through some other cell parts that we're going to end up filling some notes in here. It's a great visual. Here we have this blue part is the nucleus, and we can see something's coming out of the nucleus. That little red molecule is the mRNA. It's a copy of a gene on the DNA. And we can see here at the top right there, that little black blob there, that's a ribosome. And if you notice, the ribosomes are taking that red mRNA and they're like reading it, and then they made something blue, and they placed this blue molecule inside of these tubes, okay? So remember, the black dot is the ribosomes, and the ribosomes landed on the rough ER. This is considered the rough ER, and that's what ribosomes attach to. The ribosomes made the protein, that blue molecule, and put the blue molecule, the protein, in the rough ER. Now, the job of the rough ER is to modify the protein and send it to the Golgi. And we're going to see that here in a second. Now, this little part that breaks off here, that little circle with protein inside of it, that's called a vesicle. And that's how things are shipped around in a cell from one organelle to the next. So now we're at the Golgi, and the Golgi looks like a stack of pancakes. And the job of the Golgi is to receive proteins, modify and tag them, and send them to their final destination. And these proteins can end up in 3 locations. Okay, now I'm going to pause that real quick. In this vesicle, you see there is 2 proteins. We have the blue and we have the green. Let's take a look at 2 possible fates of proteins that are tagged and sent out by the Golgi. You're going to see that the blue ones are going to be released from the cell. So this vesicle is just going to fuse with the phospholipid bilayer of the membrane. So the blue ones got secreted or released from the cell. But you notice these green ones right here, those are proteins, and they are now stuck or embedded, or we can say inserted, into the cell membrane. We're going to talk about proteins that are in the cell membrane in the next unit, like transport proteins and receptor proteins. Well, that's how they got there, is the attached ribosome made them, sent them to the rough ER modified them, sent them to the Golgi modified and tagged them, and sent them to be inserted in that cell membrane. But we're not quite done yet, because the third destination for these proteins, they might end up being in a structure which is called a lysosome. Now, this cell just engulfed, took in that blue molecule right there, and sent it to the lysosome. The lysosome contains enzymes. If you remember, enzymes are proteins, and those proteins or those enzymes are made by those attached ribosomes. Again, send them to the RepER, which sends them to the Golgi. And watch what the Golgi is going to do. It's going to tag this molecule right here, which is an enzyme, and sends it to the lysosome. So we say that the Golgi produces lysosomes. Now, back to your note packet, and let's get some of that information down. Remember, attached ribosomes mean that they're on the RepER, and ribosomes are going to make proteins and place them in the RepER, the rough endoplasmic reticulum. The RepER is going to modify the protein and send it then to the Golgi. That Golgi is going to tag the proteins and then send them to their final destination. And since the Golgi is always receiving, tagging, and shipping out, sometimes it's referred to as the post office of the cell. So let's again review the three fates of proteins that were first assembled by those attached ribosomes. Some of the proteins can be released from the cell. That's like those protein hormones, like insulin. A cell makes the insulin, then releases it from the cell, and then it'll get in the bloodstream and travel to a different area of the body. Some of them are going to be inserted in the membrane, like transport proteins, and that's the plasma membrane or the cell membrane. And then some of them are going to end up being proteins or enzymes that are in a lysosome. That's what this next diagram is attempting to show you. You can see that we have a structure here, and it's labeled the RepER, and so that must be a ribosome. And these ribosomes are making these different proteins and putting them in the RepER. And here we see that they're now being packaged up in vesicles, and they're being sent to the Golgi. And the Golgi can modify and tag them for three different destinations. So these proteins are being released. We have that right here. These proteins are being embedded, and then these proteins are just becoming enzymes inside of a lysosome. This page is just trying to emphasize what you need to look for when you're trying to label these different cell parts. The RepER, again, is usually covered, it's going to be covered in ribosomes, and it kind of looks like a maze that you can move through. Everything's kind of connected there. The Golgi is going to look more like a stack of pancakes. Lots of times we always see like vesicles coming to and from it. So it's often diagrammed releasing vesicles from it to move to their final destinations. The lysosomes are going to look like chocolate chip cookies. And the chocolate chips are going to be the enzymes, and those are digestive enzymes that can break down molecules that the cell no longer needs, or even that a cell engulfs, like you're going to see another video here in a second. And then that cell membrane, now you kind of know what these structures are. When we draw the cell membrane, we usually show that it's got like these little things sticking out of it, and those are proteins. Oops, sorry, these are proteins, and they were once synthesized or made by those attached ribosomes. Send them to the RepER, send them to the Golgi, Golgi send them to the membrane. All right, we're going to pause here, and we're going to build some cell models.